Huh? What? All right. And action. All right, take 18. All right, so we are looking at the eyes today, and what I want to showcase first is we have a cow eyeball here, really half a cow eyeball, and you guys need to know a couple different layers. So if we were to start from the outside in, and let's imagine that these two halves were actually one complete eyeball. It was cut down the middle here, and we have then the outer, it should be transparent area here that's cut away. That would be what? The cornea. Okay, the cornea actually travels backwards into the sclera, and so cutting this in half, this would be the scleral layer here, and the function of the sclera is really just to keep form of the eyeball, right? Keep it from collapsing in on itself. All right, the other layers that you need to know are the choroid layer, and it's easiest seen as this portion here that I'm kind of literally pulling away. Okay, so it's next to the scleral region and then the choroid region there, and then you have the retinal region, or the retina here, and that's this kind of, and because this is preserved so well for so long, it's turned an opaque kind of pale here, and that's this stuff that is kind of sloughed off here. In nocturnal animals, you have the tapetum lucidum, which is kind of this iridescent covering here on the retina. Here, kind of has the blues, the reds, the yellows, and again, that's acting in essence to collect as much available light at night so that they can form an image. And so it's not necessarily night vision, but it helps because that photon is bouncing around, eliciting more of a response. Okay. What else do we need to know here? You guys do need to know the iris, which would be the colored part here, and then the ciliary body would be below that, connected in essence to the ciliary muscles that help in making the pupil smaller or larger. And the pupil would be just the open space in the middle here, right? And that's where the lens would reside as well. Here's the lens. Now again, it should be transparent, but it would fit right here. And again, it helps to bend that light, especially after the initial bending from the cornea. So it helped bend the light onto the retinal layer and the photoreceptors therein. You guys need to know, so here's the optic nerve, and it's been sliced here, but it continues right here. And here's where it inserts into the eyeball. That's known as the optic disc, okay? And the optic disc is also known as the blind spot. And it's blind spot because there's no photoreceptors there, because that's where the blood vessels and the nerve comes into the eyeball, okay? And so there's that. You do need to know that in front where the iris and the cornea would be, it's the aqueous humor. The vitreous humor has a little bit more viscous, and that's the clear stuff that you can see all up in the retina. Okay, it looks like kind of boogies. All right. So there's the eyeball. Now let's look at the cat. So I was able to get a left eyeball out, like that. And again, for many animals, not humans, we don't have a nictating membrane, but that's what this is. And you can think of a nictating membrane as actually another layer. You guys have eyelids that go over your eyeballs. They have eyelids, but they also have a nictating membrane that does the same. And you might have seen lizards do this as well. But actually, in essence, like goggles every once in a while, but also helps debris that might be floating around in the air not to get to their direct eyeball, but also cleaning that surface. So the nictating membrane is here. Then we can turn it over. And you saw me take it out of the left eye, so you already know it's a left eyeball, but on an exam, I might ask you, is this a right or left eye? My job here right now is to teach you guys how to tell. And so first we got to orient, and I'll have the muscles situated in such a way that hopefully it's easy for you guys, okay? So first we got to locate, what is this that I'm holding on to with my forceps?
Optic nerve.
That is the optic nerve, okay? So locate that, make sure you don't confuse that with a muscle, okay? And you have then, once you orient to this, you have two muscles down here and two muscles over here, all right? And then single muscles on either side. So hopefully you would know that those single muscles on either side are either the lateral or medial muscles. So first orient in that way. Now your question is it this way or is it this way? There are also subtle differences between the dorsal muscles and the ventral muscles. So in a cat, it would be dorsal and ventral. In a human, it would be superior, inferior. So make sure you make that distinction. The dorsal muscles cross pretty immediately right over top of each other, okay? Whereas the ventral muscles are about the same thickness, but don't necessarily cross immediately at the eyeball. They eventually cross, but they don't do so immediately. So that's subtle differences. The ventral muscles are here, the dorsal muscles are here, okay? Now, based on the dorsal muscles being up top and the ventral muscles being down here, you need to now locate and pinpoint the oblique muscles. The oblique name just means crossing over or angled, okay? And the oblique muscles go from lateral to medial in their orientation. This muscle is an oblique muscle. It's crossing over, going from lateral to medial. That makes this the lateral rectus, the medial rectus, the dorsal oblique, the dorsal rectus. Then we have the ventral oblique and the ventral rectus. And again, making this the left eye if you get the dorsal muscles on top, because it would orient like this in the cat. Okay? See what I did there? Adios. Any other questions?
What are cataracts?
Cataracts. Okay, so that's basically, this guy has a cataract now because the portions of the inside the lens is a flexible material, but also not completely solid. Mm-hmm. And so you have the chemical structure breaking down and not maintaining its clearness. It's hardening and that sort of thing. And so it's,
to get rid of that, they put like an artificial lens in?
They could do that. They could also, use a laser to break up the crystalline forms that are becoming crystallized, in the lens itself. Mm-hmm. Yeah. So, cool.